Welcome to Wildcat Science with Mrs. Haugo. This is biology, and we are in the cell biology notes, so this is cell biology notes part 2. In the previous video, we learned about the cell cycle and why cells divide, and the two parts are interphase and mitosis, and we need checkpoints to make sure that cells don't get cancer. And the phases of interphase are G1, S, and G2. And there are a couple of checkpoints in there. And we learned the other major stage of the cell cycle is mitosis, and it is divided into prophase, metaphase, anaphase, telophase, and cytokinesis. We know that some cells divide more quickly than others. And we also learned about activity number 14 and stem cell differentiation, how some cells are stem cells and some cells are differentiated or specialized cells. And we talked about how that happens, where in a specialized cell, some of the DNA gets turned on and some of the DNA gets turned off. Then we learned about different types of stem cells, like totipotent that can make any form of cell, and multipotent that can make only a couple of types of cells, and specialized cells, which can only be that one thing. And we talked about some misconceptions about stem cell research, and we talked about ethical versus scientific questions. Now today we're talking about activity number 16, HIV/AIDS infection and cell organelles. So please start a new page or skip 3 lines and write that title. Also, I would like you to write the challenge. How does HIV take over a cell's structures and organelles during infection and use them to reproduce? In class, we're going to be reading page 244, and we're going to talk about how viruses are not like animal cells, they're not like plant cells, they're not like bacteria cells, they are totally different. So please write all this down. So a virus is made of a protein coat and then either DNA or RNA inside. And so a virus is different than any other living thing because it cannot reproduce on its own. It must infect another cell to be able to reproduce. There are 2 types of cells. You don't have to write this down, but do pay attention. There's a lytic cycle, where the virus takes over the cell organelles and makes many copies of itself, and then it actually bursts open and releases the viruses, and that cell is now dead, and it'll go and infect other cells. There is the lysogenic cycle, where it actually the virus puts itself into your DNA, and now it's part of your DNA, and every time your DNA gets replicated, the virus gets replicated and makes the viral DNA gets replicated, and it makes more and more viruses. So those cells aren't going to burst open. So we're going to quickly watch an animation about that, about how HIV infects your other cells.
The human immunodeficiency virus, or HIV, destroys cells of the immune system, making the body vulnerable to a host of diseases. HIV has surface proteins called GP120 that attach to cells that have CD4 receptors on their surfaces. CD4 is found on the immune system's helper T lymphocytes and on scavenger cells called macrophages. The binding to CD4 and other cell surface proteins called co-receptors, not shown here, allows the virus to fuse with the cell. The lipid membrane of the virus incorporates into the cell's membrane, while the viral core enters the host cell. During entry, the core uncoats, allowing the viral genetic material, which is RNA, to enter the cytoplasm. In addition to RNA, the virus carries enzymes that participate in the first few stages of viral replication. Before the virus can commandeer the cell to make more viruses, viral enzymes must first convert the viral RNA into DNA. The viral enzyme reverse transcriptase copies the RNA into complementary DNA, or cDNA. Reverse transcriptase has a high error rate and frequently leaves mutations in the copied DNA. The mutations result in variant forms of HIV, which allow HIV to evolve quickly. The viral RNA is destroyed by another viral enzyme called ribonuclease H. Reverse transcriptase then synthesizes a second DNA strand, using the first strand as a template. The double-stranded viral DNA enters the nucleus with several types of viral proteins, including an enzyme called integrase. Integrase enzymes splice the viral DNA into the host cell's chromosomal DNA. The integrated viral DNA is known as a provirus. The synthesis of the viral genome begins with the transcription of the proviral DNA into RNA. The viral RNA contains the code to produce the capsid proteins and other proteins needed for viral assembly. In spliced form, the RNA also codes for the envelope proteins and other auxiliary proteins. Envelope proteins are produced in the rough endoplasmic reticulum, ER, and then move through the Golgi apparatus before arriving at the cell surface. Full-length viral RNA molecules, along with all the other viral components, assemble at the membrane and bud off from the host cell. Although it's not clear exactly how HIV curtails the immune response or causes destruction of helper T cells, the viral infection ultimately results in loss of virtually all functional helper T cells in the immune system.
Okay, we'll talk more about that video in class. So we will have somebody be able to answer the challenge and make sure our vocab is done. And we have another case study, the rotavirus case study, that we will be doing with our post notes and disease information worksheet. Okay, activity number 17. Just a preview here, and we'll be going more in depth later. So activity number 17, disease interventions. We're going to be doing the vocab. We're going to be looking at the challenge. So please write down World Health and Sustainability and diseases. So you're going to be in charge of one of these diseases. Please write all 6 of them down. Cervical cancer, diabetes, HIV/AIDS, malaria, rotavirus, and tuberculosis. So we're going to figure out with these 6 major diseases, what kind of interventions can we do? Like how can we help? So that's what we'll be involved in with activity 17 and 18 coming up. But before we do that, we are going to be doing a review and a quiz on these notes. All right. Good job, everybody.